Hello friends, I am Gaurav Mukwana. I welcome you all to my another video tutorial Biotech guide. In previous video lecture series, we have discussed on amino acid, protein, carbohydrates. Lipid is remaining in biochemistry. So in today's video lecture, we are going to discuss about lipid. So in this video lecture, we are going to discuss about a classification chart and brief discussed about on each type of lipid with the example. So stay tuned with us and enjoy this video lecture. Let's start first with introduction. What are the lipids? So lipids are defined as esters of alcohol and the fatty acid. We'll take one example. Likewise, triglyceride formation. So triglyceride is mainly formed by glycerol and fatty acid molecule. What happens here? Hydroxyl group of fatty acid will combine with a hydrogen ion of a glycerol and form a water molecule which is released, and linkages form in between of a glycerol and the fatty acid molecule C bond O bond C, which is known as a ester, and the process is also known as the esterification. Okay, so that's why the lipid are also defined as esters of alcohol and the fatty acid. Now what are the properties are there? They are insoluble in the water and soluble in the non-polar organic solvents. They are mainly vitamin A, D, E, K, those are the lipid soluble vitamin. So this is the simplest introduction of the lipid. Now we'll go through classification chart. So basically lipid are classified mainly into three types: simple lipid, then compound lipid, and a derived lipid. So simple lipid again classified into two: triglyceride and the wax. Another one, compound lipid, which is further classified into three: phospholipid, glycolipid, and the lipoprotein. And the third one, derived lipid, which is not a type, but we can say component of the lipid is also known as a derived lipid, which is mainly again combined and the form a lipid molecule. So those are fatty acids, steroids, then eicosanoids, and the glycerol. Let us discuss one by one. First, simple lipid. Simple lipid again classified into two: triglyceride and the wax. First, triglyceride. As we discussed in definition, triglyceride is the consist of a glycerol molecule bonded with a 3 fatty acid through a ester linkages. Glycerol is a 3 carbon alcohol with 3 hydroxyl group, as you can see here, and the fatty acid is a long hydrocarbon chain with a carboxyl group at the one end. Okay, so this is the triglyceride formation, also known as a esterification process. Now what is the function? So basically energy storage and the energy production are the two main function. Primary function is to energy storage in adipose tissue, and the broken down through hydrolysis, triglyceride release the fatty acid which can be used by cells for energy production. Next, second type of simple lipid is wax. So wax is a nothing but one molecule is changed. Likewise in triglyceride, as we see glycerol and the fatty acid, here glycerol is replaced to the alcohol section. Alcohol section, that means a long chain of alcohol. One thing you have to remember that hydroxyl group of fatty acid and hydrogen ion of any other molecule, likewise here one alcohol is combined and ester linkage is formed in between of a 2 carbon atom of these two separating molecule. Okay, so esters of fatty acid with a long chain of alcohol form a waxes contain a fatty acid plus cetyl alcohol. Here one example is there. Likewise we'll see example plant waxes, which is a cutin, which is a outer layer of a plant cells. Then bee waxes is yellow and soft, secreted by wax glands of a worker bees. Then air wax, cerumen, which protects the air membrane. Then sebum, secreted by a sebaceous gland, which is mainly present on the face. Then linolene is a wool wax. Now second we are going to discuss about a compound lipid. So compound lipid again classified into three types, mainly phospholipid, then another one is a glycolipid, and the third one is a lipoprotein. We'll discuss two example of each type. First is phospholipid. So structure consists of a glycerol, two fatty acid and the one phosphate group. Okay, again glycerol, fatty acid and a one phosphate group. That's why the name suggests phospholipid. And a polar head group such as a choline, ethanolamine, serine or inositol. Mainly based on this polar head group, again phospholipid classified. We'll see later. Now let's focus on function. So major components of a cell membrane forming the lipid bilayer, they are also involved in a signaling pathway and a membrane fluidity. So first type of phospholipid is what? Phosphatidylcholine. Structure is glycerol backbone, two fatty acid tail, phosphate group and the choline is there. That's why the name suggests phosphatidylcholine. Function is major component of a cell membrane involved in a membrane fluidity and the structure integrity. And the sources found in the egg yolk, soybean and other plant and animal tissues. Second subtype is phosphatidylethanolamine. As name suggests, lethanolamine. Okay, so here one polar group is ethanolamine and the components are same: phosphate group, two fatty acid and the glycerol backbone. Function is plays a role in a membrane fusion and the curvature of biological membranes prevalent in the nervous system. Sources found in the bacterial membranes and in the inner leaflet of a eukaryotic membrane. Second type of compound lipid is glycolipid, which consists mainly a carbohydrate group with a lipid molecule. Okay. Function is mainly play roles in a cell recognition, signaling and the stability of a cell membrane, and they are also abundant in a nerve tissues and the cell membrane. Subtype: first cerebrosides and the second one is a ganglioside. So first cerebroside. Structure consists mainly composed of a ceramide group. Okay, that's why the name suggests as cerebrosides. Ceramide is a fatty acid linked to a sphingosine molecule also, and a single sugar residue such as a glucose. If glucose is present, so also known as a glucocerebrosides, and the galactose is present, so they are also known as a galactocerebrosides. Function is mainly important in nerve cell membrane and a brain tissue involved in a cell signaling and the interaction. Sources found primarily in the nervous system, particularly in a myelin sheath of a nerve cell. So this is the important cerebrosides, which is mainly present in the myelin sheath of a nerve cell. Then second subtypes of a glycolipid is a gangliosides. Structure containing mainly complex glycolipid containing ceramide backbone attached to a oligosaccharides. What's the main difference? Here mainly ceramide is combined with two monosaccharide, mainly galactoside and the glucose, and what here in gangliosides mainly backbone, ceramide backbone is attached with the oligosaccharide chain, and a structure is known as a gangliosides. Now the function play a critical role in a cell recognition, signal transduction and the cell-cell interaction, also involved in a neurological development and a function. Source is abundant in the nervous system, especially in the brain. Then another third type of compound lipid is lipoprotein. Mainly when lipid combined with a protein is known as a lipoprotein. Function is mainly transport lipid in the blood and the lymphatic system. Example include low density lipoprotein and the high density lipoprotein. First, low density lipoprotein. Density is higher, smaller size than VLDL and the ideal. VLDL stands for a very low density lipoprotein and the ideal stands for intermediate density lipoprotein. Composition is high cholesterol content and the lower triglyceride content. Function is deliver cholesterol to cell throughout the body. That's why known as a bad cholesterol because of their association with atherosclerosis. Atherosclerosis is a deficiency where mainly lipid molecule is trapped in the arteries. Okay. Then second subtype of lipoprotein is high density lipoprotein. The density is highest and the composition consists high protein content, moderate amount of phospholipid and the cholesterol, and the major function is collect the cholesterol from the tissues and the transport it back to the liver for excretion and the recycling, often referred to as a good cholesterol due to its role in reducing atherosclerosis risk. Okay, so mainly we will differentiate with the LDL and the HDL through their function and their mechanism. Now third type is derived lipid. So basically when we deassociate all the lipid molecule, a primary component will achieve is known as a derived lipid. Likewise, fatty acids, steroids, eicosanoids and the glycerol. Okay, so derived lipids are the smaller molecule derived from the larger lipid compound through various metabolic processes or a chemical modification. They include mainly fatty acid which we'll discuss already, and also we're going to discuss in the next video lecture briefly. They are also known as the simplest form of a lipid, consisting of a long hydrocarbon chain with a carboxylic acid group at one end. They can be saturated, no double bonds, or unsaturated, consist of one or more double bonds. Then the steroids. Steroid are the one kind of a lipid with a characteristic structure of a 4 fused carbon ring. Example include cholesterol, which is essential for cell membrane structure and the synthesis of steroid hormones like estrogen and testosterone. They have a cyclopentanoperhydrophenanthrin nucleus made up of 4 rings A, B, C and D. The ring A, B, C are a cyclohexane and the D is a cyclopentane. Then eicosanoids. Signaling molecule derived from a polyunsaturated fatty acid. They include prostaglandins, then thromboxane and the leukotrienes, which regulate inflammation, blood clotting and a smooth muscle contraction. Then fourth, glycerol, a 3-carbon molecule that serves as a backbone for a triglyceride, storage lipid and the phospholipid, structural lipid. So this is all about of a derived lipid. So here we completed our topic. I hope you enjoying very well and got it very well. Thanks for watching, and if you want to get more and more interesting video like that, so join with us and be a perfect biotechnologist.